Hello everyone and welcome back. Dear students, we are going to discuss the structural details of seeds in angiosperms. A seed is defined as a fertilized and metamorphosed ovule containing an embryo enclosed in resistant protective coats. You can also say a seed is the initial dormant stage of diploid sporophytic generation in the life cycle of spermatophytes or phanerogames. Spermatophytes or phanerogames are those plants in which the sex organs are exposed and they produce seeds. Okay, seed is the product of fertilization. That means it is formed as a result of fertilization between male and female gametes. That is why we say seed represents the dormant stage of diploid sporophytic generation. Okay. Now some important structural details of a dicotyledonous seed. First of all, its covering, which is known as seed coat. Uh, there are two coverings in seed coat: the outer thick and resistant layer, which is called as testa, and the inner thin and membranous layer called as tegumen. Okay. A seed is attached to the inner wall of fruit by a small stalk called funicle at a point called hilum. Okay. It is a stalk-like structure which attaches the seed to the wall of the ovary at a point, okay, called as hilum. The point where the funicle fixes attaches to the seed, this is known as hilum. We will see it in the next slide. A small pore is present close to the hilum in the testa, which is known as micropyle, and a ridge may be seen in continuation with the hilum on testa, which is known as raphe. In some cases, in some seeds, the hilum is extended in the form of a ridge-like structure, which is known as raphe. And enclosed within the seed coat, there is an embryo consisting of embryo axis, which is known as tigellum in botanical terminology, and there are two cotyledons. All the contents which are enclosed within the seed coat, they are together known as kernel. The embryonal axis consists of two ends, the radicle and the pilumule. Here you can see. This is the lateral view of a seed. This scar-like structure is the point where the funicle attaches to the seed. Okay, and close to this hilum, you have a pore-like structure in the testa, which is known as micropyle, and this micropyle is very much important because it helps us in the entry of oxygen inside. Okay, it also helps us in the absorption of water so that the seed can germinate. This one represents the image if a seed is cut open, and you can easily see the structural details of the embryo when you cut a seed open. Here you can see you are having an embryonal axis, and the embryonal axis contains two ends. This one is the radicle, which forms the future root, and this one is pilumule, which forms the future shoot. Okay, and the point of attachment of besides this, you have two cotyledons attached to the embryonal axis, and these two cotyledons they store food in X albuminous seeds, and they do not store food in albuminous seeds. That we will come to know in the later slide. Now, other minute structural details of the seeds of dicotyledons. The cotyledons are attached to the embryo axis at the region which represents the first node of embryo. The first node of embryo represents the region at which two cotyledons emerge. And the part of the embryo axis between radicle and first node is called hypocotyle. The region between the radicle, okay, between the radicle and the first node, that means the point of attachment of two cotyledons, is known as hypocotyle, while as the part between pilumule, I mean future shoot, and the cotyledons is called the epicotyle. Okay, and most of the dicotyledon seeds are called X albuminous or non-endospermic, a majority of the dicotyledon seeds are X-albuminous, or you can say non-endospermic, because they lack the endosperm at maturity. That means in these seeds, the endosperm is consumed in entirety. Okay, all the endosperm is consumed by the developing embryo. In such cases, entire endosperm is consumed. That I have already told you. A few dicotyledon seeds, a few species of dicotyledons are there, like castor, we have castor. They possess endosperm at maturity and are known as albuminous or endospermic seeds. Okay? In albuminous seeds, the cotyledons are thin and papery, while as in X-albuminous seeds, the cotyledons are thick and fleshy because they store the necessary food material. Okay? जो albuminous seeds होते हैं उनमें जो cotyledons होते हैं, they are very much thin and papery because they do not need to store the food because they possess a lot of endosperm. While as in case of X-albuminous seeds, since they do not possess the albumin, they do not possess the endosperm, they need to store the food material in their cotyledons. That's why their cotyledons are fleshy. Okay, this is all about the structural details of dicotyledon seeds. I hope you all understand this. Thank you and God bless you all.